Hi guys, today we're going to talk about chapter 17, which is the process of evolution, and so this will be uploaded for you guys on Blackboard so that you can watch it at your leisure. Okay, so your book starts off talking about what are called antibiotic superbug farms, and so antibiotics are used to fight bacterial diseases. Okay, we use them for pathogenic organisms like E. coli or for salmonella, any of those nasty little bugs that can cause you to get sick. And so, way back, probably in, I think it was like the 60s and 70s, people started using antibiotics just as a regular thing in their organisms on their farms, like cattle, pigs, poultry, fish, to promote growth. And this basically set the stage for like all these crazy antibiotic-resistant bacteria. Remember that bacteria are living, and so when you put them in an environment where they need to either change or die, they change because they're living. They can adapt and they can grow in their environment. So the agricultural industry is like a huge user of antibiotics because it makes the chickens grow faster. You can get more meat production. The downfall of this, though, is that we're producing all of these organisms that have these superbug colonies living inside of them. Because bacteria are small and very short-lived, they can evolve really fast because their lifespan is only a couple of days, and so they can become resistant to antibiotics given that their environment is rich in antibiotics, because they have to either, again, evolve or they have to be the fittest to survive, as it were. And so if they don't survive, then they die. And again, bacteria are living organisms. They can evolve. They can be naturally selected for resistance to antibiotics. So we started practicing treating livestock with antibiotics because it guarantees that we're going to get a bigger yield of organisms like chickens or pigs or whatever. They don't get sick as often because you're using antibiotics on them constantly, but this is really bad for the meat, and it also guarantees the production of antibiotic-resistant bacterial strains in those populations. And so in 2013, there was an investigation done and where they went to several different grocery stores in several different states, and they grabbed samples of chicken meat, and then they tested it for antibiotic-resistant bacteria, and over half of those samples were positive for those antibiotic-resistant bacteria. So developing new antibiotic drugs takes a really long time. It's difficult to do, and it's a lot slower process than the bacteria can evolve. And so superbugs do create a threat for humans. If you catch something like MRSA, for example, which is a Staphylococcus aureus bacteria that has a methicillin resistance, we might not be able to treat it. And so coming up with these new antibiotics is incredibly important, but then also limiting the use of antibiotics when they're not like totally life-saving and necessary is also very important, because we want to stop giving these things the environment for them to reproduce and then also to evolve these resistance genes. So the first thing I want to tell you guys about is that individuals, like on their own, don't evolve, but populations do. And so a population, if you guys remember from the beginning of the semester, is just a group of interbreeding organisms of the same species at the same time in the same place. Oh, excuse me. So they have different alleles for the same genes. Okay, so all of these individuals are going to have shared genes, like genes for coat color or genes for how to make an antelope or whatever, but they're going to have different alleles. And so this basis of variation, this is the basis of variation within a population. And so if you've got organisms with two distinct forms of a gene, they are called dimorphic. And so, for example, if you've got purple and white flower colors in pea plants, that's a dimorphic trait. Two different kinds. And so polymorphic traits means that you've got more than two distinct forms. Probably the best example of this is blood type in humans, because you've got A, you've got B, you've got AB, and you've got O. So mutation, we talked about a little bit before, is the raw input for evolution and natural selection in the wild. And so like having those little tiny mutations, most of the time those mutations are either going to be silent or they're not going to be advantageous, but every once in a while you have a mutation that is advantageous, and it does give the bearer or the person or the organism that has those mutations an edge over everybody else. And so every once in a while those mutations are really good. So other events that shuffle alleles into different combinations, like genetic drift or gene flow, any of that kind of stuff, kind of like helps to influx new alleles from different populations into a population of organisms in another place. So there are several different types of mutations. So neutral mutations are going to be like your silent mutations where you just have no effect. So like you might have a mutation for something that doesn't do anything for your fitness. It doesn't do anything for the population as a whole. A lethal mutation is going to result in death. And again, that's a lot of mutations are lethal or they're not advantageous. But then you also have beneficial mutations. And so this bestows a slight advantage. So we've got a little bit of an edge towards survival or reproduction. And so if that happens to an organism, they now have a slight edge on everybody else. Like maybe they're a little bit taller so they can reach the fruit in the trees a little better, or maybe they're a little more brightly colored and so the females find that a little bit more attractive. Those individuals are going to get to breed more, and they're also going to get to pass down those mutations to their offspring. And so that's how you get like a new influx into a population. Okay. So when you're looking at a population and you're kind of wondering like what changes are going on, it's important to note the allele frequencies. And so that's the actual number of alleles for a gene that you have in a population at a time. So the gene pool is just all the genes in a population all taken together all at the same time. And the allele frequency refers to the abundance of a particular allele, so maybe like blue-eyed allele or something like that, among the individuals of a population. So if you were looking at Americans, for example, like if we had, I don't know, maybe just like, you know, a sample size of 100 people in Kansas City, and you wanted to look at the allele frequency for blue eyes, you would have to go in there, you'd have to find all the people that had blue eyes, you'd have to find all the people whose parents had blue eyes to find out if anybody was a carrier, and then once you've got those numbers, you can add them up and you can actually assign a numerical value to how many, like literally how many alleles do we have in this population for blue eyes. So microevolution is evolution on a very small scale. So this is change in allele frequency in the gene pool of a population or species. This is constantly occurring because you've got alleles coming in from different populations, just like, for example, if you had a population of, I don't know, like squirrels or something living in your backyard, and then there were squirrels from 10 miles away that migrated over here from a different population, they're going to come over and they're going to interbreed with the squirrels that live in your backyard. So that's changing the gene pool a little bit because you've got new individuals coming over and breeding with the current individuals. And so that's microevolution. So Hardy and Weinberg, they were two scientists that applied probability rules to population genetics, and so their work is actually very useful and very helpful today when we're looking at allele frequencies in populations. And so their equation is P plus Q equals 1, or P squared plus 2PQ plus Q squared equals 1, and we'll talk more about that here in a second. So genetic equilibrium is one of the things that Hardy and Weinberg looked at very closely. This just means that your genetics are in a stable state. They're not really moving around very much. The allele frequencies are going to remain constant from generation to generation. And this is a theoretical model that we use to kind of use as a base jumping point for looking at allele frequencies in populations, because this does not occur in nature. There are a lot of stipulations for Hardy and Weinberg's equilibrium in order for it to work, and one of those things is random mating. Like you can't have males fighting over females, and you can't have females specifically picking a male over anybody else. There can be no genetic drift, there can be no gene flow, on and on and on. And so this is a theoretical thing. It doesn't really occur in nature because you're not going to find no gene flow, no non-random mating and whatever in a natural setting. So the conditions for that genetic equilibrium is that mutations can never occur because we have to keep our allele frequencies constant. The population has to be infinitely large, which is another thing that like it doesn't happen. You can't have an infinitely large population. The population is isolated from all other populations, and that means that there can be no genetic drift and no gene flow. Mating has to be random, and all individuals survive and produce the same number of offspring. So that way all of the allele frequencies stay the same. And so again, in nature this is never completely true. Oops. So Hardy-Weinberg, they take this concept of that genetic equilibrium under ideal conditions, and you can use that again. You can use this, this model to kind of show what allele frequencies are doing in a population. But again, this is theoretical. So this kind of shows you what Hardy and Weinberg's equilibrium looks like. And so again, their equilibrium, their equilibrium looks like this. So we've got our P squared up here, which stands for your homozygous dominant individuals. You've got your 2pq, which is your heterozygous individuals, and you've also got your q squared, which is your homozygous recessive. And so this formula is p squared plus 2pq plus q squared equals 1, and that 1 represents 100%. So again, this is used as a benchmark, and it's used to kind of like look at how populations are changing in a specific place at a specific time. But again, this is theoretical. You can't use this forever because those rules for Hardy-Weinberg would never be completely satisfied all the time. And so for this one, we're trying to determine the carrier frequency of an allele that causes a genetic disorder called hemochromatosis. And so the allele frequency among people with Irish ancestry is about 14%. So that means that your recessive allele, your Q, and also remember the other part of their equation is that Q plus P equals 1. Okay, so Q is 14% of the population. So this is the allele frequency of people with Irish descent. And so that means 100, or 1 minus 0.14 is going to give you 0.86. That's where this number comes from. So 14% of the people have this have this allele. 86% of the population have this allele. And so then you just multiply them together. 2pq equals 0.24. So these are going to be your carriers that you've got in the population. So 2 times p, 0.14, times q, 0.86, is going to give you 0.24. Okay, so let's look at some patterns of natural selection and like what that does in a population. I'm going to just kind of skip through this here real quick and show you guys pictures of what this is. But directional selection just is when you've got one extreme phenotype that's favored. Stabilizing selection is when you've got an intermediate phenotype that's favored. And then disruptive selection is where you've got two phenotypes that are selected, and they're kind of like on either end of the spectrum. And so this is directional selection, where actually this is not directional selection, this is directional selection. Going back this way, so directional selection where you've got an extreme phenotype that's favored. Stabilizing selection is where you've got one intermediate phenotype that's favored, and then disruptive selection is down here where you've got two extreme phenotypes that are selected for in an environment. And I'll give you guys examples in here in just a second of what that looks like. So again, directional selection looks like where you've got one extreme phenotype that's favored over another. And so let me show you. So there it is again. There's your directional selection right here. And so this is what that looks like, where you've got one extreme phenotype that's favored. A really good example of this is the peppered moths in England. And so this is where peppered moths used to mostly look like this. But then when the Industrial Revolution hit and we started putting a lot of soot and stuff in the atmosphere, and we started putting a lot of like burning coal and getting a lot of like black icky stuff, it stuck to a lot of their trees. And so even though this used to be the wild type, this used to be the kind that you would find most likely in the environment, because if you look over here, there's that white guy. It blends in a lot better with that white birch tree background than this dark one does. But however, when you had all that soot and stuff sticking to all the trees around the time of the Industrial Revolution, now those little white guys stick out like a sore thumb, and they are much more easily picked off by predators, and so this became the new wild type. And so that's the directional selection, is that now that white phenotype is a lot less frequent than the darker phenotype, and so now that darker extreme phenotype is selected for. Stabilizing selection is where you've got an intermediate phenotype that is favored. So not too big, not too small, just right in the middle. A really good example of this, so on here they've got these little nuthatches, and so the really small birds don't do very well, the really big birds don't do very well. This kind of like middle body mass size seems to do the best. This is also true for like human birth weight. Little tiny itty bitty babies don't tend to do very well, and really big babies also don't tend to do very well because they can't really get out of their mother at the time of delivery. And so having a baby that's around like 7 pounds is better than having a baby that's like 4 pounds, which is too small, or 13 pounds, which you might not have been able to get out of you way back in like the 1800s before they had cesarean sections. And so that's stabilizing selection, when you've got an intermediate phenotype that's selected for. And then disruptive selection is where you've got two extreme phenotypes that are favored, and then the intermediate phenotype is not favored. And so the example they give you in your book here is these black-bellied seed crackers. So let me show you a picture of these guys. And so what they were looking at here was beak depth, and so they were looking at the actual distance between here and here. And so the ones that have the smaller bill size tend to do okay because they can they can feed on a certain type of seed or a certain type of food, and the ones that have the bigger bills that are 15 millimeters wide also tend to do okay. The ones that were in the middle, the between 13 and 14 millimeter range, didn't tend to do very well because their beaks were too big to crack all the little baby seeds that these guys would work on, and they were too small to get the really big seeds that these guys would work on. And so that intermediate phenotype is not selected for in this example. Okay, so some of the things that can bring up our diversity, because again, remember, being a little bit diverse and having a little bit of differences between other individuals is really super important, because if there's an ecological disaster, if you